DNA sequencing is a process where we determine and identify every single DNA base, every single DNA element that is in the genome of an individual. And there are 6 billion of those in every cell, in every normal cell, in every person. And when we apply DNA sequencing, in the Cancer Project, we first figure out what those 6 billion DNA bases are in the normal cells in that person, and then we take some of the tumour in that person and figure out what the DNA bases are in the tumour.
In a cancer, the DNA is changed in many different ways. If you think of your DNA sequences as a book, the way you can change is that you can lose a whole paragraph in it, like a deletion, or you can duplicate a paragraph or a page multiple times, or you can have a misspelling, a single letter that's been changed, or pieces that gone to the wrong place. The page, the pages fall apart and you put it back together in the wrong order. And any of those disruptions will lead to misread when the cell proliferate, and that leads to mistake and alter behavior of the cell. And that ultimately, when you have enough of those things go wrong, you become cancerous.
In a typical genome, we might see 30 sites that differ between a person's normal genome and their tumor genome. But we look very closely at these 30 differences to try to figure out which ones are the meaningful ones. And one way we do that is to look across many people's tumors. And so we say, is this gene hit very often? So we had to use DNA sequencing at large scale for this type of project because the point here is to really do a systematic survey across many genomes. We can look at a small number of samples and be able to make guesses and say, well, this one might be a recurrent change, this one might be important. But to get down to the rare events, which might lead us to new areas of therapeutics or new understandings of why a tumor arises, we have to be able to find events that occur only at a 3% level or a 5% level. And unless you're studying hundreds of samples, you're never going to find those events. You just simply don't have the statistical power to find them.
We're looking forward to the $1,000 genome or the $100 genome so that we can do these analyses on tens of thousands of samples, and we can follow tumors not just once, but look at the DNA sequence of the tumor as it responds to therapy, and look to see how it's changing, and we can adapt our therapeutic regimens as the tumor evolves.
Sequence information is going to be used at all different levels. So there will be some who will just use the very end point of this project, which is the knowledge of which particular genes can be important in which particular cancer. And that can spawn a whole career of research into the mechanism of what's going on in that cancer. Others will want to look at every single base in the particular disease, every single DNA letter in that particular disease that they're studying, because they'll want to know all of the subtleties that can occur under all of the different circumstances in that disease. So there's a generality of how things will be used, but there's one thing that'll be common to everybody who's either studying cancer or treating cancer, and that is that they will have to become versant in the language of DNA.